First of all, I would like to thank the organizers for the opportunity to present our work. I'm Zhe Ji. I'm an assistant professor at Northwestern University in USA. So today I will talk about our recent work to use ribosome profiling technology to identify genome-wide stable non-canonical peptides. Based on the conventional genome annotation, there are four types of transcripts encoded by the human genome, including the mRNAs, that's protein coding genes, and the long non-coding RNAs, pseudogenes, and small non-coding RNAs. However, during the history, it has been a challenge to separate mRNAs versus link RNAs and define actively translated over linear frame. So with the advance of deep sequencing technology, so scientists have identified many novel transcripts from the human genome. So, but how can we say, okay, one transcript is protein coding or it's non-coding? So there are two commonly used approach. One is to use an arbitrary cutoff, like on 100 amino acid, to distinguish mRNA and link RNA. For the transcript with a overlinear frame longer than 100 amino acid, people say, oh, this is protein coding. If it is there is no long or a conserved overlinear frame, then people say, oh, this is a link RNA. So currently, there are some computational methods developed to calculate the coding potential of the transcripts. So these computational methods were trained based on the annotated protein coding genes with like medium length of 500 amino acid and conserved during evolution. Although those methods can identify functional proteins, but it can ignore those stably folded macroproteins or species-specific peptides. And it's known that there are actually many functional macroproteins in our genome, like IPL41, SLN, and APELA, right? So they are all pretty short. So to address this issue, first of all, we need to basically identify all the possible translated over linear frames across all the transcripts, and then to say, oh, what are the over linear frame can encode stable functional proteins. To address this issue, we need to go back to the fundamentals of RNA translation. So basically, 40S ribosome recognize 5' cap structure and start to scan until it meets the start codon, that is ATG or one nucleotide variant. At the start codon, 60S ribosome will join, forming this 80S ribosome, and this 80S ribosome moves one codon per step to decode over linear frame into protein. So the ribosome profiling technology is the state-of-the-art method to study active translation. So that's invented by Nico Ingoglia and Jonas Weisman. So they use the RS1 to digest the lysed cell and isolate this ADS ribosome complex and recover the RNA footprints for deep sequencing. So it has been widely applied to study different aspects of translation. So when I was a postdoc, so basically I analyzed ribosome profiling data. If we do the data analysis carefully, so basically we can see clear, strong 3-nucleotide periodicity of the redistribution across actively translated over linear frame that starting from the start codon and end at the stop codon. And actually we can observe clear 3-nucleotide periodicity for individual codons of actively translated over linear frame. So basically, 3-nucleotide periodicity represents active translation. And then we basically developed a software called RibORF. It's a logistic regression classifier to identify genome-wide translated over linear frames. So basically, what we do is select the candidate over linear frames with a start codon ATG or one nucleotide variant and a stop codon in all transcripts. And then select the expressed ones. And for the expressed ones, we check whether there is strong 3-nucleotide periodicity that uniformly distributed from the start to stop codon. So we use the fraction of the reads in the first nucleotides of the codon to model the 3-nucleotide periodicity and use the percentage maximum entropy value to measure the uniformness of the redistribution across the candidate ARF. So basically, for the actively translated overlapping frame, they have high fraction of the reads in the first nucleotide and also high PME value. We can see many of these link RNA and 5' TR are actively translated. So it can achieve high accuracy to identify a genome-wide translated ARF with AUC greater than 0.99. So recently we applied this approach to nearly all the published ribosome profiling data from yeast to human and select this high quality data set with 3 nucleotide periodicity across the ARFs to annotate genome-wide translated ARFs across those species. So in the end, we found that in humans, basically the annotated overlinear frames only account for like 40% of all actively translated ARFs. So there are a lot of translated ARFs in 5' TR, called UARFs, overlapping UARFs, internal ARFs, downstream ARFs in 3' TR, and many of these ARFs in long non-coding RNAs and pseudogenes. So you can see the mammalian translaton is much more complex than yeast. So there are about 60% of the 5' TRs are translated, and 20% of 3' TRs are translated. So then the question is like whether the peptides generated by these non-canonical ARFs are functional or stable or not, right? For that question, so first we need to consider the length of these non-canonical peptides. So the median length of this, for example, UARF peptide is about 15 amino acid. If we think about it, it's very, very short. It's probably not functional. But there are actually some outliers here that are pretty long, actually longer than 100 amino acid. For example, this UARF peptide generated by this protein coding gene actually is 106 amino acid. So during the past few years, we and others basically identified these genome-wide translated overlinear frames that open up to the possibility to tackle their functional roles in the cell. And there are a few important papers published. So basically, for example, this one, so basically we collaborate with Todd Golub's lab at the Broad Institute and performed the large-scale overexpression of these non-canonical peptides, and found that many of them, like link RNA encoded non-canonical peptides, are stable in the cell. And we performed the CRISPR screening experiment and found that actually knocking out many of these non-canonical overlinear frames actually inhibit cell proliferation. And this work from Jonathan Weissman's lab basically took a similar approach and reached the similar conclusion that basically to say many non-canonical peptides are stable and knocking out can affect cancer cell survival. And this work is from my postdoc mentor, Aviv Regev, and Cassie Wu's lab. So basically, they used our software and then annotated this genome-wide translated ARFs and then used this MHC found immunopeptidome database, the mass spec data basically to search this unannotated protein database, and found many of these non-canonical peptides actually are presented by MHC. The number is close to 2,000 of those non-canonical peptides encoded by 5' TRs or link RNAs. And this work is from the Claude Perot and Jonathan Yediel's lab at NIH. So basically, they found that many of those non-canonical proteins either can be found in the cellular proteome or be found in immunopeptidome. And they seem to be mutually exclusive. So the emergent theme is that many of these non-canonical proteins probably unstable. They're degraded but can function as MHC. But there are some of those non-canonical peptides actually can be stably expressed in the cell and can have functional roles. So recently we asked the question, how can we computationally distinguish non-canonical ORFs encoding stable peptides versus quickly degraded byproducts? So to address this question, so basically we collected 343 stable macroproteins that are shorter than 100 amino acid, those from RefSeq defined database, and this John Presner's work. So they did like overexpression with these hundreds of link RNAs and found some are stable. And this is from Jonathan Wiseman's paper. So, the John Presner's papers basically also found 100 of those macroproteins are basically are undetectable from epitopic expression, because those stable and unstable peptides were like their expression was driven by the same 5' TR, 3' TR, so these undetectable macroproteins probably are unstable ones. So those peptides were used as the negative. So basically, we want to examine our features that classifying these stable versus undetectable macroproteins. So first, we check the conservation levels of those peptide sequences during evolution. We use this follow CSF score measuring the peptide conservation across mammals, and found these stable macroproteins tend to be more conserved than these undetectable ones. And then we group these stable peptides based on their conservation levels and check their peptide length, and found that these peptides basically show comparable length regardless of their conservation levels, and they are all longer than these undetectable peptides. So this data suggests that actually maybe the conservation doesn't matter so much. But the length actually matters more. And most of these non-canonical stable macroproteins are longer than 60 amino acids. So then we go back to the question, why people use 100 amino acid at the beginning to define as a cutoff to define the stable functional protein. There are some pre-assumption made. So basically, one assumption is like long peptides are likely to be folded into secondary structure and to be stable. And the other assumption is long opening frames would not emerge by chance during evolution. So basically, we randomized the genome or transcriptome sequence of this macroprotein encoding transcripts. For those macroproteins, their transcript median length is about 1 kb, and at 2% FDR, the length is about 70 amino acids. That is much shorter than 100 amino acids. Actually, the majority of those non-canonical stable macroproteins, actually they are longer than expected by chance if we consider their transcripts actually are shorter. On the other hand, these stable macroproteins tend to encode a domain, for example, P-farm domain. Then, based on this conservation, length FTR, and the domain, we build a logistic regression model called PEP-score that can classify these stable versus undetectable proteins. So that PEP-score has a good performance classifying the two groups of macroproteins with AUROC equal 0.94. And from this logistic regression model, we found actually this log length FTR actually contribute most significantly to the classification with most significant p-value, but less about the conservation level. So then we apply our PEP-score to all the open-ending frames identified from the ribosome profiling. We found that about 13% of these non-canonical peptides show high PEP-score, and about 80% show low PEP-score, means that vast majority of these non-canonical peptides probably are degraded. But there are 13% actually have high PEP-score, and those peptides from 5' TR, like URFs, overlapping URFs, like internal RFs, DRFs are 3' TR, and pseudogene and link RNAs. And the median length of this non-canonical peptide is about 90 amino acid. And generally, they are poorly conserved during evolution with this negative follow CSF score. So the data indicated that most of these non-canonical peptides with high pep scores show longer length but low conservation across mammals, indicating that the existence of a large number of the species-specific young proteins. So basically, they're just longer, but they are not conserved across mammals. To further validate our pep score, basically we fused this ORF sequence with a flag tag and then the same 5' TR and a 3' TR sequence. We performed this epitopic expression of this ORF sequences. We found that some of these non-canonical peptides are quite stable, like SLC35A4. Its expression is comparable to GFP. This is as a positive control. So, but some of them actually are not so stable, like CDH8, the expression level is pretty low, but actually many actually are like empty vector. We cannot see anything if we do a transient of expression about 100-fold. So there is a correlation between this peptide expression versus this pep score. We selected this about 25 this URF encoded peptides that's encoded by our 5' TR. For the high pep score ones, some of them actually are pretty stable. So basically, the expression level are nearly comparable to the GFP. But for some high versus to medium pep score peptides, their expression level is not so stable. So then we think why their expression level is lower. It's not like they are not translated because their expression was driven by the same 5' TR, 3' TR versus those stable ones. So basically, we then treat the cells with proteasome lysosome inhibitors, and we see that the inhibition of proteasome lysosome pathway, we can stabilize some of the peptides with moderate to high pep scores, but cannot rescue the expression of those very short URF peptide with low pep scores. So suggesting that these very short ones are probably just degraded by cellular protease. They don't need like proteasome lysosomal pathway to fragment them into small pieces. Then we pick some of the high pep score and this stable peptides for further functional study. For example, this peptide is encoded by the 5' TR of SLC35A4. We performed the expression of those 4-length transcript, including the URF sequence with a flag tag and the MERF sequence with HA tag. And we can see that mutation of this URF start codon will diminish the expression of the flag tag and increase the MERF protein expression indicated by the HA tag. And the immunostaining experiment showed that this URF peptide is localized to the mitochondrial outer membrane protein, and do a twofold overexpression can slow down cell proliferation and damage mitochondrial membrane potential. So we think this URF peptide is a mitochondrial membrane protein, regulates cell proliferation and mitochondrial membrane potential. So there is another internal ORF encoded peptide. So basically, the start codon ATG is the downstream of this canonical start codon. This peptide is about 100 amino acid long. So the co-IP mass spec experiment show that this peptide interact with the mitochondrial processing peptides, and the immunostaining experiment show that this peptide is localized to mitochondria in the membrane. And we show actually the mitochondrial peptides can cleave here to generate 11-mer like C-terminus fragment that can be translocated to the cell membrane. And the twofold overexpression of this internal ORF peptide can activate some development gene and suppress some immune response gene. So taken together, I think stable macroproteins can be generated by the 5'-TR, 3'-TR overlapping coding region and annotated non-coding RNAs of our genome. And the longer non-canonical peptides, typically longer than 60 amino acids, are likely to be functional and stable. But the short ones can, even they are degraded, they can function as a new antigens for disease therapy. So we think this is a conventional evolution approach to create species-specific proteins. So basically, different species can leverage their existing mRNA structure and enhancers to produce this non-canonical peptides that's basically show some species-specific expression. But finally, I would like to emphasize this functional protein of this link RNA doesn't contradictory to their RNA function. So basically, a link RNA can function through the RNA domain in the nucleus while they can generate some functional peptide in the cytosol, so they are not contradictory. Finally, I would like to thank my lab members who did the work, especially Haiwan Yang and Qianru Li, and my funding sources. Thank you very much for your attention.